Hi, this is going to be a study session, and I am just trying to learn basic biology. This particular session would focus on cytology, which is part of human anatomy. More specifically, though, this video would focus on cell structure or cell membrane. This particular video and the tone of the video should be very relaxed. I just want to get into the habit of speaking and making study session contents like this without fear of any setbacks, such as like what would people think of this and stuff like that. So that is not part of the uh, worry list for me today. Today, I just want to be able to go through this particular set of videos. Not videos, but this particular set of videos. There's two videos in particular, and I will walk through how I got to this position, which particular videos where I following, and most of the video should focus on reading this, which is an extensive read on the structure of the cell membrane. So that should be the core focus. I also am going to take help from ChatGPT, Google Gemini in this case, and it's supposedly okay. Let me just read this. It was generating something and then it stopped. I don't know what happened. Still responding somehow for some reason. I need to, I guess, stop responding. Reload page. Okay, let's see if this works. Okay, cool. Let it respond, and in the meanwhile, let me walk you through the um, table of contents for today. So, what I know so far, I knew very little going into cell membranes. I mean, obviously, I had the biology classes in my school days and college days, but I never quite focused, I never quite grasped the core essence of biology and medicine in general or human anatomy. But now it seems like it would be worthwhile to study or learn about these things. So I think it was the main reason I got to YouTube, and this person, Professor Fink, uh, his lecture series is something I really found I found it very helpful. So this is why I'm following his lecture series here, and the basic review of anatomy and cytology, introduction, etc., etc. I think there is two or three more videos. There are two or three prior videos to this, and I have been enjoying his lectures. Very informative in terms of how he delivers his lectures, especially the handwritten notes that he puts in a projector, and he basically projects his own handwritten notes, it seems, which is really uh, good and helps me see the world of biology or human anatomy in his through his lenses, which is great. Number two, he has a great sense of humor, and he presents with a great, like, he's very engaging in his way of delivery, so that is fun to watch as well. This is, by the way, part two. I have seen part one, and part one really got me into wanting to learn more about cell cytology or like human anatomy in general, precisely because of the distinction it drew between like how medicine works. So, for instance, I think if my take is correct from part one, how the medicines generally work is because not because is the following way. For instance, our cell has some protein receptors, and it basically signals you what type of cell this is. And sometimes, not sometimes, but most all the time, I think medicines or drugs are used to attract, or not attract, but track down that particular receptor. It's like, okay, this is how we identify that particular cell. If your like stomach hurts, medicines would be made thinking of that stomach cell, right? And that stomach cells, set of cells, could be identified because that those particular those particular cells would have a unique identifier of some sort. So that's how drugs can be targeted. And the issue here is that, and this is why side effects exist, because those same identifiers could be found in cells in, let's say, your head. So the common example, or the good example he used, is like every time we have a cold, run your nose, we take drugs and we feel dizzy because the same receptors are affected in the head as they are in treating cold. So that was something really like, it simplified medicine, biology, and human anatomy to me in a way that made me feel like, okay, I can study more on this, and by doing so, I might even be able to grasp a few of the things. So, with that being said, that was my um, initiation into wanting to read more or learn more about this. Now, what do I know about, in particular, about cell membrane so far? And before I get into that, I think it would be better to precaution the viewers or listeners, whoever it may be, that even if I'm wrong on certain things, I think, especially like on the outgo as I'm describing them, there are ample ways I can rectify that through following the video. So whatever I say now, don't take that as a face value. Keep in mind that I'm only learning, and I'm going to have a better grasp at the end of it than I have currently. So my current views can be flawed or subject to criticism, so should not be taken as a medical advice of any sort by any means. Having said that, I think okay. Let me run down what I know so far about cell and cell membranes. Okay, what I know so far is that, I think I'm going to use this. This is much more interactive. Cell structures. So if this is a typical cell, we have the outer layer, which is a membrane, and cell membrane. Let me see if sense of the environment. Okay, sure, why not? Meta and okay, so I think this is like different um, levels of evolution of a cell, like cells divide, etc. As cells grow and divide, the meiosis and mitosis phase, etc. But in any case, what I know so far about cell structures is the following. I think this was it. Okay, the following. So it's made of phospholipid bilayer. So there is a layer. This particular thing is basically a layer, and it is mosaic in that it doesn't only have lipids, which is like phospholipids. It also has proteins, like either serving as a direct tunnel between the outer world and the inner world, or like proteins sticking out of the structure. And one of the core reasons why that is the case is because of the nature of lipid with respect to water. So lipid is not it doesn't go, it is hydrophobic, in other words. And that is the reason why water or water like substance cannot get directly into the cell through lipid. And there is a good reason for that. I think it doesn't require any explanation, but I'm going to go ahead anyway. Because if like water like substance, which is like most of our body, could get into the cell, it would mean that our cells are not permeable at all. Like our cells are semi-permeable, but if water could get in, like every content of the cell would just like wither away, like escape, or there wouldn't be any need for a structure to begin with, right? So water-like substances are separated basically by this phospholipid bilayer, and uh, but then the question arises, like what's up with substances that are water-like, and what about them? How would they come in and go? So that is where protein comes in. Like remember I said the protein channel, there exist protein channels. Is there mitotubules protein? No, there's not, but there are other visual places where like it shows, for instance.
Globular proteins, which are peripheral, associated with the surface of the cell, may be enzymes or glycoproteins. Other integral proteins serve as carbohydrate globular collectively transport fibrous proteins may span the entire membrane. Layers of phospholipid bilayer the cell membrane is mainly.
It had a meltdown. Okay. I'm going to shut it all the way down. Shut it all the way down. Okay. Construction of the cell membrane. This is like the protein channel I was talking about. These are like the sticking out aspects of carbohydrates, and these probably form the signaling unique identifier stuff, saying that like, okay, I am this cell, I am that cell, to drugs and etc. And there are also proteins on the outer surface, and we're going to learn more about that. But the main thing is that certain water soluble substance like glucose, amino acid, and ions, and water most importantly, would come in through, I think if I'm not wrong, through this channel, and that is the key takeaway. And other lipid like substances can go through like this lipid bilayer chain, which is like this one is a phosphate, and like it goes into it.
In this activity,
Oh,
you
no.
will learn through building.
Okay, this is a phospholipid. So the the phosphate head, the round head is very much, it doesn't like, yeah, it likes water. That's why it's like headed on this side. On the outer side it's headed out, and in the inner side it's headed in. Like it's like, you get the point. So they are attracted to water, but the middle portion isn't, and as a result things cannot get in or get out, except if the things are lipid structure or lipid soluble constructs like oxygen and carbon dioxide and steroid. So I think it would be helpful to understand what is a steroid, by the way. What is a steroid? Steroid is an organic compound with four fused rings arranged in a specific. No, okay, not this. I think I'm not going to get into it right now. I'll let this portion help me with the specific details of it. But this is where we are currently, and now begins the official study session for me. Official. Now begins the study segment of the video, where I basically read into most of the things that are said with respect to this video part 2, and specifically about ion channels and like nature of proteins and what they're like. I think I'm not going to play the entirety of this video, or not even like some portion of it. I don't want to like steal from this video. It's just here to showcase that I'm following this lecture series. So if you're inclined to do the same, you can do so, and I will link this channel and this particular video in my channel description. Having said that, let's read the Google entries, not Gemini entries rather. So Gemini is a um, Gemini is a place I start with the basic cursory stuff, so it helps me get into, like, it helps me ease into the thread or topic that I want to learn, which is a great thing. And here it is. I want to know about cell membrane and plasma membrane, like what is it, what how is it made of, what is the structure, what function does it serve, and like are there any other names it came up with this. The plasma membrane, also known as the cell membrane, is a vital component of every human cell. It acts as an outer body separating cell's internal environment, cytoplasm, from the external surrounding. Both internal and external surroundings are water-like substance. And cytoplasm, I think, is just protein plus water. That probably is the reason why it is jelly-like. And I got that from Professor Fink. Structure. The plasma membrane is particularly composed of a phospholipid bilayer. Imagine two layers of phospholipid molecules arranged back to back. Each phospholipid has two main parts. Number one, hydrophilic, water-loving head, which is basically the phosphate part. This part contains phosphate groups and faces outwards, okay, towards the water environment, both sides, cytoplasm and outside, which is the cell. Hydrophobic, water-fearing tails. These are made of fatty acid chains and face inwards, forming the core of the membrane. So what is a fatty acid is going to be another thing that I need to learn. So there are like two things, I'm going to note them down. Fatty acids, and which one was the other one? The other one was steroid. This arrangement creates a selective permeable barrier, and selective permeable barrier is a good phrase I should use, meaning it only allows certain substances to pass through while keeping others in or out. Beyond the phospholipid bilayer, the cell membrane is a fluid mosaic of other molecules, including proteins, and we're going to learn more about proteins just in this thread. Like what specific functions does it serve? I think I'm going to couple that with the visual aids. This channel website has great visual hints to make you understand or aware of how the things work. For instance, this is a phospholipid. This is a fibrous protein that I think it says at the outset that there are like two regions or sets of proteins that we need to contend with. One is a fibrous protein, which is this. The other one is a like integral or peripheral protein. So I probably need to know what does a fibrous protein do, and what does the integral or peripheral type of protein do. So integral would be, I think, glycoprotein, and pore protein, I can see H2O molecules like directly going here. And this probably serves as a receptor or a unique identifier. And channel protein is probably peripheral. Is it peripheral? I need to understand like what is the distinction with respect to one being integral and one being channel protein. I hopefully the response that it generated in like part 3, like the third question, answers that. Does it? It so far facilitate diffusion. Okay, I think it didn't, so let me re-ask this. Can you explain me? Can you explain to me the difference between fibrous fibrous protein and integral and feral proteins with respect to cell membrane. I want to know what glycoprotein pro which type of protein glycoprotein and glycoprotein, I think, and glyco pore and channel proteins are. Okay, let you respond. Okay, I think we were here. Hydrophobic water fearing tail. These are made of fatty acid chains and face inwards, forming the core of the membrane. Core of the membrane, okay. They're forming the core of the membrane. That is a good way, apt way of putting it as well. This arrangement creates a selective permeable barrier, meaning it only allows certain substances to pass through while keeping others in or out. Beyond the phospholipid bilayer, the cell membrane is a fluid mosaic of other molecules, including proteins. These are embedded within the lipid bilayer, integral proteins, or attached to its surface, peripheral proteins. Okay, it does answer that. Okay, they perform a wide range of functions, including transport, signaling, and cell recognition. So we are looking at three distinct or key functions that proteins serve. One is um, transporting. It like transfers what? It transports substances. There's also the issue of signaling. So signaling has to do with, I think there's also like the sentence that I was originally going to say, was that signaling also deals with neurotransmitters and enzymes slash hormones. So we have to be aware of those things as well. Forgetting what they are, I think Professor Fink like kind of he gave a good explanation, and this is like one of the other things why I came to like, or came to be interested enough, came to be more interested to learn about human anatomy. Is he basically drew the distinction between an enzyme, a hormone, a like vitamin and minerals, etc. So probably the distinction between vitamins and minerals is that one is organic, the other one is not. And neurotransmitters is probably, what is neurotransmitters? It probably is the stimulus that we face. For instance, like if we put our hands on a hot stove, it probably sends signal directly to our brain, and that probably is something. And then what else? Hormones are enzymes, are basically catalysts, and he is going to always make this sound. He's going to like tap his hands together to signal the thing that it catalyzes, or sets the reaction in motion. So stuff like that, cool stuff. So that is probably signaling. And cell recognition is unique identifier. Signaling and cell recognition is not the same thing probably. Okay, so what is the difference between the two, you ask? The difference, I think, would be the difference would be like our body does a better job. So our body naturally does a better job of sending or allocating resources to, not allocating resources, but allocating certain substances or ingredients to specific cells, because it does so uh, by design, by nature. I would say by design, I think would be too much religious, but that is the core function it serves, right? So when we need when we need a particular set of hormone, it basically sets those particular set of hormones to uh, certain particular cells. And then cell recognition has to do with medicines that we take that are not organic to our body. It's like not part of our body. We are taking it, intaking it, and that's why there is a need for self recognition. And there's a types specific types of cells do specific things. I think Professor Fink elaborates on that. Anyhow, some integral proteins are transmembrane proteins spanning the entire membrane. This would be like one of those sentences that I'll have to let go. I don't want to delve too much time learning about this right now. Cholesterol. This lipid helps maintain mm, the membrane's fluidity and stability, preventing it from becoming too rigid or too fluid at different temperatures. So this is the third thing that I want to know more about: cholesterol, cholesterol, lipid. Now the cholesterol test is a lipid profile test. So learning more about this would be fun. Then we have carbohydrates. These are often attached to proteins forming glycoproteins or lipids forming glycolipids on the outer surface of the membrane. They collectively form the glyco glycocalyx. I didn't want to mess up the pronunciation, which plays crucial role in cell recognition, in which plays a crucial role in cell recognition and adhesion formation. The components of cell membrane, especially lipids and proteins, are synthesized and assembled through the cell's endomembrane system. Okay, let me read that again. Formation: the components of the cell membrane, especially lipids and proteins, are synthesized and assembled through the cell's endomembrane system. Okay, I need to understand endomembrane system. This is another taking note. This is like fourth on the list. Endomembrane system. I need to understand how these proteins and lipids are originated and how are they developed, which includes the endoplasmic reticulum and Golgi apparatus. If I'm not mistaken, I think Professor Fink, uh, in the third part of this um, specific review of cytology in his lecture series, goes into Golgi body. I think he starts with Golgi body, or the reticulum that he mentioned. What was the name? Reticulum, endoplastic reticulum. So I'm not going to jump too far ahead. I'm just going to rely on him to convey that information to me. Phospholipids, for instance, are primarily synthesized in the endoplastic reticulum. Okay, proteins are also synthesized in the endoplastic reticulum and processed and shorted in the Golgi apparatus before being inserted into the membrane. Okay, let me read that again. So the phospholipids, the circular head, for instance, are primarily synthesized in the endoplastic reticulum, which I don't know what it is. I know it's part of a cell structure, but I don't know. I don't remember actually. I don't recall specifically where it sits or is situated. Proteins are also synthesized in the endoplastic reticulum and then processed and shorted in the Golgi apparatus before being inserted into the membrane. So that is where Golgi apparatus comes in. Like it um, processes, I don't know what processing a protein would mean, and shorted in the Golgi apparatus. The fluid mosaic model proposed by Singer and Nicholson in the 1970s best describes the structure and the dynamic nature of the plasma membrane, where its components are constantly moving and rearranging. Functions: the plasma membrane performs several critical functions for the cell. Number one is selective barrier. Number two is protection and support, transport. Proteins embedded in the membrane act as channels, carriers, and pump to facilitate the movement of specific ions, nutrients, and other molecules across the cell membrane. Cell communication and recognition. Proteins and carbohydrates on the outer surface act as receptors for hormones, neurotransmitters, and other signaling molecules, enabling cells to communicate with each other and respond to their environment. They also serve as identity markers, allowing the immune system to distinguish between self and non-self. Identity markers is also crucial because white cells would attack them based on if they recognize those particular cells to be part of the body as opposed to not part of the body. Adhesion. Certain membrane protein facilitate cell to cell adhesion, allowing cells to form tissues and organs. Signal transduction. I don't know what the word transduction means. The membrane can generate signals in response to external stimuli, leading to changes within the cell. This should be interesting, but I'm not going to get into it right now. Then I asked, explain to me phospholipid bilayer and simplify how it's made of and what does it do. The phospholipid bilayer is a fundamental structure of the cell membrane, acting like a selective skin around every cell. What it is a two-layered fence made of tiny identical bricks. Each brick in this fence is a phospholipid molecule. We have the thing that we mentioned about water, one part liking water, the other part not liking water, and this like two the tails go together and the heads are basically outward out of the cell and within the cell. How is it made? Simplified. Building blocks, think of it. Cell assembly. Once enough phospholipids are made, they don't need much help to form the bilayer. Because of the unique love water and hate water parts, they automatically arrange themselves into two layered structures. When placed in a watery environment, it's like oil and water separating naturally. Insertion. As new phospholipids are made, they are inserted into the existing membrane, allowing the cell membrane to grow and repair itself. Let me read that again. Insertion. As new phospholipids are made, they are inserted into the existing membrane, allowing the cell membrane to grow and repair itself. Okay, so this is how the repairment, like organs of the cell probably die continuously and have to be replaced continuously as well. So like as we eat, as we do things, we constantly hopefully regenerate different aspects of our body, even in the micro level. So while the cell actively makes the individual phospholipid molecules, the bilayer structure itself largely forms spontaneously due to the chemical properties of these molecules in a watery environment. Okay, what does it do? The phospholipid bilayers main job is to act as a gatekeeper for the cell. Think of it as a very smart filter. Okay, filter is such a great way to explain it. I like gatekeeper is kind of a strong word, bounded your barrier, fence, etc. But filter is a great way to put it. Gives the inside in and the outside out, selectively permeable. Most important function because it's hydrophobic, water hating core. The bilayer allows only certain things to pass through easily. Small uncharged molecules like oxygen, carbon dioxide can slip through. Okay, small and uncharged. So apparently carbon dioxide and oxygen molecules are small, and uncharged, yes, uncharged, there's no charge potential attached to it, but I didn't know it was small. They were small. Large molecules, charged molecules or ions like sugar, salt, water molecules generally cannot pass directly through the lipid bilayer. This is where the proteins embedded in the membrane come to help transport them. Okay, forms the fundamental, stable, selective, permeable backbone of the cell membrane, allowing the cell to control its internal environment and interact with its surrounding in a regulated way. Cool. These are good. Should I take a water break? Sure, why not? I don't have to pause it, just going to keep commenting on it. Um, Let me comment on how am I liking my study sessions so far. I think this is a good system. It's so much relaxed, and I'm just talking to myself, explaining things to myself, and reading the things that I wouldn't read otherwise. So number three is explain to me what protein does, like what protein does what functions in details with respect to cell membranes. Should I read this here or I should start with this? Can you explain to me the difference between fibrous protein and integral and peripheral protein with respect to cell membrane? It's great that you're dividing into the details. You're diving into the details of great cell membrane proteins. There's a crucial distinction between how proteins are classified by their overall shape slash solubility, fibrous versus globular. So I think globular, this one. So this is like the globular. Yes, took me a while. But this is like the globe-like structure, but it's like as the thing passes through, like this portion is closed. As it goes, yes, and it like closes down. I don't think I'm going to be able to explain that verbally, but that is the gist. And how they are classified by their association with the membrane, integral versus peripheral. Okay, so we are fundamentally dealing with fibrous and globular type of protein, and then the integral versus peripheral distinction only comes in as a direct thing for the membrane. Like it only becomes a thing when we are taking it to, like, this particular different definition of integral versus peripheral is only contingent on where these proteins are situated, but fibrous versus globular protein probably serve different purposes, uh, regardless of where they're situated on the cell, right? Could it be the case? Let's break it down. Fibrous protein versus integral versus peripheral proteins. Fibrous proteins, a classification based on overall protein structure slash shape definition. These proteins are typically elongated, strand-like, sheet-like. They are generally insoluble in water, definitely, and play structural roles. They often form long fibers or sheets that provide strength, elasticity, and support. Example would be collagen in connective tissues, keratin in hair and nails, actin in muscle cells. Okay, collagen, 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 collagen. Collagen. I have heard of collagen, especially in respect to connective tissue. What is a connective tissue? A connective tissue is probably, I'm thinking muscle, like joints and stuff, but also I'm kind of conflicted about the definition and wonder if this is not talking about blood here. What is a connective tissue? What while googling what a connective tissue? Tissue that supports, protects, and gives structure to other tissues and organs in the body. Connective tissues also store fat, helps move nutrients and other substances between tissues and organs, and helps repair damaged tissues. Connective tissues is made up of cells, fibers, and gel-like substance. Trim the skin and any traces of connective tissues from the sides of the, what the hell is blood a connective tissue? Yes, blood is considered a specialized connective tissue. It consists of red blood cells, white blood cells, platelets, and plasma, all super suspended in a fluid matrix. This fluid matrix, the plasma, is an extracellular matrix, making blood unique among connective tissues. Okay. So I am vindicated after all, after being wrong 50%. Relevance to cell membrane. While fibrous proteins can be associated with cell membrane, e.g., proteins of a cytoskeleton that attach to the membrane for structural support, they're not typically embedded within the membrane itself in a way integral proteins are. Instead, they usually form networks underneath the membrane, cytoskeletal protein, on outside the cell, extracellular matrix proteins. What? This portion I definitely do not get. Definitely do not get. While fibrous proteins can be associated with the cell membrane, proteins of the skeleton that attach to the membranes for structural support, they're not typically embedded within the protein itself and the way integral proteins are. Okay, so was it just delineating the different protein types? They're like, these are different types of molecules of the cell membrane: phospholipids and fibrous proteins. Here are some examples of globular shaped proteins: glycoprotein, pore protein, and channel protein. Now there's the fibrous protein may span the entire membrane and serve as receptors for the cell. What does a receptor do? Receptor probably serves to attach with other cells. So for instance, I think I'm going to write it down: receptors. Receptors and connectors, are they the same thing? That would be something hopefully this video, this this interactive Q&A bot can answer, right? Instead, they usually form networks underneath the membrane, cytoskeletal proteins, and outside the cell, extracellular matrix proteins. Integral membrane proteins, a classification based on membrane association. Definition: these proteins are primarily associated with the cell membrane. They're embedded within the lipid bilayer, meaning they have hydrophobic regions that interact directly with the hydrophobic tails of the phospholipids. Okay, hydrophobic. How they're removed: you can't easily remove them by simply changing salt connection. You can easily remove them by simply changing salt concentration or pH. You need to disrupt the lipid bilayer itself, typically using detergents like which act like soap, uh, breaking up the fatty membrane. Wait, what? um, Using detergent. So is it like the fat, the slimy fat that we get eating like beef and stuff, right? Is this it? Types: transmembrane proteins. These spans the entire lipid bilayer, which with parts exposed on both the side and the outside of the cell. Many receptors, channels, and pumps are transmembrane. Span the entire thing. Okay. Integral monotopic proteins. These are embedded in only one leaflet half of the lipid bilayer and do not span the entire membrane. Okay, I guess. Functions: They are workhorses of the membrane involved in transport, signal reception, cell adhesion, and enzymatic activity. I think another break would be necessary. I can sense my voice not uh, being as okay as it was at the start of this thing, but okay, I'll try to push forward. These are the workhorses of the membrane involved in transport. Transport, I understand. Signal, like, okay, I am this cell. Signal reception, okay. So, signal reception would be something like